Dear viewers, welcome to the Poultry World webinar on bacterial chondronecrosis and osteomyelitis and the antibiotic-free strategies to prevent this costly disorder. Together with Luis Valenzuela of BIOMIN and Professor Robert Wideman of the University of Arkansas, we will take an in-depth look in the causes of BCO lameness and share the results of scientific research which has demonstrated how better gut performance can support flock profitability without antibiotics. Keeping the birds able to move without restrictions or pain will keep them on the feed and on the water, and that's, as you know, essential for performance. My name is Fabian Brocketer and I'm the editor of Poultry World and the host of this webinar. This is a truly interactive webinar. I would like to point out to you the possibility to ask questions to our speakers via the chat function in the lower right-hand corner of your screen. This chat function will be live during the whole webinar and will be used later on during the question and answer session. Furthermore, you can respond to poll questions several times throughout the session. Let me introduce the first speaker of today, which is Luis Valenzuela. He is Product Manager Microbiomes at BIOMIN for the Poultry Star product line. He holds a bachelor's degree in animal production at the National Pingtung University of Science and Technology in Taiwan and a master's in fish nutrition at the National Taiwan Ocean University. In addition, Luis holds a degree from the Specialized School of Engineering in El Salvador as a technical industrial engineer. Before I give the word to Luis, I want to pose the first poll question. I will start the poll. The poll question is antibiotic reduction an objective in your poultry operation? You can press the answers by A is yes, B is no, and C, you're not sure. I see the first results already coming in at my screen. I will just wait for a moment for the last poll votes to roll in. Yeah, it's still moving a little bit around, so I will stop the poll and show you the results. Display. It seems like 97% of all voters sees antibiotic reduction as an important agenda point. Does this surprise you, Luis?
Oh, good afternoon. Thank you, Fabian, for your introduction. Professor Wideman, good afternoon, dear audience. Well, it's not a surprise. We have been visiting customers, and the first thing they said is that they would like to have a antibiotic-free solution so they can use in their flocks throughout the production cycle. So yeah, we agree with this.
No big surprises there.
Yes, no.
Luis, BIOMIN has continued to deliver a national non-antibiotic feeding solutions for the poultry industry for over 3 decades. Based on that experience, what are some of the keys to reducing antibiotic usage in modern poultry?
Okay, before I give you the question, the answer to the question, I just would like to remark that we are not against the total use of antibiotics. However, we know that they are there for a reason, and we should leave the antibiotics for what they are. Now that I tell you this, BIOMIN has a 360-degree approach when it comes to antibiotic reduction programs, and it's because we have to combine a proper nutrition, we have to strengthen biosecurity, we have to have the right strategy among the veterinarians and nutritionists, where, for example, vaccines can be used and additives in a proper way, but at the same time we have to also have good practices in the farm. So that's the way we can do it. And why we need to do all this? Well, we have to remember what is the importance of the healthy gut. First, the healthy gut, well, the gut, the gastrointestinal tract, is the largest organ in the body. this is the interface, as you can read from your screen, between the bird's immune system and the environment. This, at the same time, is responsible for the breakdown of feed and absorption of nutrients. And why is all this? And because we have to know that a healthy gut or gut integrity is the backbone of performance, and that's what you can see why you have that picture at the end. Now, BIOMIN has come up with a toolkit. In this toolkit, we have a portfolio of products which are in two groups: gut performance management and mycotoxin risk management. In gut performance, we have products such as organic acid, phytogenics, and of course probiotics. And in mycotoxins, we have mycotoxins the activator, which is what we are known for, which is Mycofix or Biofix in another parts of the world. So, yes.
Can you tell us about probiotics? What's available commercially, and how can probiotics support gut health and performance?
Okay, yes. If we move on, well, here I can give you a little bit of a background to understand how is a microbiota inhabitant in an adult bird. Now, this is 10 to the power of 13th of viable bacteria per gram of digester. This one keep changing from day 1 up on the 30 days of age. So is highly dependent on stages of the production, like pre-starters, starters, grower, finishers. All these, they will have a fluctuation in their microbiota. And therefore, a rapid establishment of a beneficial gut microflora or microbiota as early as possible is essential. So we can have a stronger birds and we can stabilize as soon as possible the microbiota. So you asked me what probiotics are there. Well, we can categorize probiotics in, well, first we have prebiotics, which is a feed for the probiotics, then we have the nutrition, for the probiotics, and then we have single-strain probiotics, multi-strain probiotics, symbiotic products, which is a combination of a probiotic with a prebiotic. We have undefined culture products, and we have multi-species symbiotic. As you see in your screen, we have Poultry in the multi-species symbiotic, because besides the probiotic, we have the prebiotic. Currently in the market, the most commercial products are bacillus-based products. However, these are not 100% effective for the gut immunomodulation, which is what we target with Poultry, to keep a gut good integrity. Now, good gut integrity, I'm not saying they don't work. They have a purpose, but for gut immunomodulation, that's basically what we promote as a direct effect. Now, how did this come up? This product had an extensive background, an extensive research of 8 years of international development in Europe, where we isolated more than 400 strains from the gut of healthy chicks through different production cycles. And then we had to evaluate through in vitro trials and in vivo trials which ones were the best ones, the best candidates. And from this, we narrowed down to at least to have the main drivers of gut health. The main drivers in this case for us are Enterococcus, Bifidobacterium, and Lactobacillus. This is a composition that you will see in Poultry. And I'm not saying these are all, but here are the main drivers of gut health. If you can see in your screen, they are from different sections. You have from the jejunum, you have from the ileum, and you have from the cecum. They have main effects in different areas. That's why we have them. The Enterococcus will protect partially, will create a double layer on the epithelium. Therefore, we will have the competitive exclusion, what probiotics are known for. We have the Bifidobacterium animalis, which is there for priming the immune system or triggering the immune effect. We have the Lactobacillus, which, well, it will secrete lactic acid and we will create the proper environment. All of them secrete lactic acid, and they will create a proper environment just for the beneficial microbiota to be there in the receptors and have the intimate interaction between the enterocytes and the microbiota. Now, in this composition, we also have, as I said before, the fructooligosaccharides. This one is there for a purpose, and the purpose is to boost the action and the growth of Bifidobacterium, because as we already know, in humans is similar, this one grows the slowest. So we needed to keep up the pace with the other strains that are in the product, and for this reason, we have to boost them a little bit or stimulate the growth with fructooligosaccharides, which is isolated from inulin. So for this, we have Poultry. is a multi-species host-specific symbiotic designed to improve gut health and to make day-old chicks and birds of all ages more resistant to pathogens. We are proud to say that this is the only multi-species probiotic, which is, well, symbiotic, that is registered in the EU. And this is good. Why? And people sometimes ask us why you have to mention this. And the reason is because this is like a benchmark for good quality. So it's a good quality product that has been extensively investigated to see if it's safe for humans and for animals. And it is. We have proven that. And because of this reason, we had this, as you can see in your screen, you have this label that says Innovation Award from the Eurotier winner 2016. Among 25 innovations, we're proud to say that Poultry Star was the only feed additive that was awarded in that conference or exhibition. Then you can also see there that says Biotaglic. That's we are certified for organic use as well. So yeah, that's what I can tell you from Poultry Star.
Well, thank you very much, Luis, for these insights in probiotics and Poultry Star in specific. Luis will be available for the question and answer session. So please don't forget to type in your questions to Luis in the chat box. We will answer them later. That brings us to the second speaker of today. It's an honor for me to introduce Professor Robert Wideman. Dr. Wideman began his career at Penn State University, where he rose to the rank of Professor of Poultry Science. In 1993, Professor Wideman was recruited as the Distinguished Professor of Poultry Science and Arkansas Poultry Federation Chair at the University of Arkansas. He served as a professor and associate director of the Center of Excellence of Poultry Science. Professor Wideman recently retired as an emeritus professor. So glad you could join us, Professor.
Thank you.
Can you explain to me and our viewers, of course, what BCO Lameness is and how it develops?
Sure. Okay. And where is it? Okay. Okay. So what is BCO? BCO is an acronym for Bacterial chondronecrosis with osteomyelitis. It basically means that there's damage to the growth plates in the long bones of the leg and the flexible thoracic vertebrae, and we'll show you some of that later. And this is causing these cells in the growth plates to die and form wound sites, which then become infected with bacteria, and then the bacteria infect the bone as well, which is osteomyelitis. This is a common cause of lameness in the United States and the European Union. It typically affects 1.5% of the broilers, usually starting at about 30 days of age, but it can start earlier than that. And really, we've had outbreaks that are epidemic outbreaks that have caused lameness in over 15% of the flock. We call this a metabolic disease because it's definitely linked to fast growth. Basically, these birds are growing so rapidly that a subset of them, their skeletons simply can't support the weight, and particularly the soft cartilage growth plates can't support the weight. This slide shows the likely spots where we're going to see most of the BCO lesions. This includes the proximal head of the femur, the proximal head of the tibia, and the flexible thoracic vertebrae. These are sites of intense mechanical torque and stress on the bones, and this is where we begin to develop microfractures in the growth plates. These microfractures are called osteochondrotic microfractures, which simply mean they're fractures within the cartilage layers. This diagram is designed to show us the progression of this problem, or the pathogenesis of this problem. And this is our working hypothesis, but through our research, we've confirmed virtually every stage of what you're going to see on this diagram. We start out here at the end of the long bones of the legs, or the growth plates in the flexible thoracic vertebrae. And with fast growth, these growth plates become thick and fragile, and the weight of the bird can cause cleft formation or microfracture formation. And these are wound sites, but if they don't get infected, the birds simply continue to grow, and they don't seem to exhibit any untoward issues. Back in the 80s, over 40% of the broilers and many of the turkeys were shown to have these microfractures. Recent evidence indicates that over 70% of them, this occurs in over 70% of the birds. Where is the source of bacterial infection? Well, bacteria can quickly or easily, relatively easily, leak through the intestinal epithelium, and as long as the immune system is competent and this rate of leakage is relatively slow, then that can be accommodated by the body's defense systems and no bacteria break into the bloodstream. However, under conditions of stress, which opens up channels between the epithelial cells, between the tight junctions, and stress which inhibits the immune system, floods of bacteria can enter the circulation, circulate throughout the body. We call this polymicrobial bacteremia, and then circulate to the blood vessels that are supplying the growth plates. At these blood vessels that supply the growth plates, the terminal capillaries have incomplete vascular walls. We call this a fenestrated endothelium. Cellular elements and bacteria that are circulating in the blood can exit the bloodstream at this point, and if they find a wound site, the wound site activates the pathogens to become pathogenic, and they start an infection. This slide shows a typical infection in the proximal end of the tibia. Basically, here is the growth plate, and right here beneath the growth plate, we see an osteochondrotic microfracture. We see by the change in the color, the sort of cheesy color here, that this is infected with bacteria. And here we see a bacterial sequestrum surrounded probably by a biofilm to protect it against the immune system. So this is where the problem starts, and at this stage, the bird may not exhibit specific lameness, but here we can see that this has grown into a big bacterial abscess, and these birds would be unable to walk and would be removed from the flock and humanely euthanized. And so this is the progression of what we're seeing.
Professor, can antibiotics be used to reduce this condition, and what are the disadvantages?
That's a great question. So obviously this is a bacterial infection, and in fact, if we go to the literature, we see multiple species of pathogens that have been isolated from these lesions. We see Staphylococcus, we see Enterococcus, such as Enterococcus cecorum, we see other species, and we even see some Salmonella species that are isolated here. These have been identified by swabbing the samples and culturing them on culture medium. But if we use more sophisticated genomic techniques, such as have been used by my colleagues at the University of Arkansas in Dr. Youngmin Kwan's lab, we can find evidence that dozens, if not hundreds, of bacterial species are present in these lesions. Two of these species, Staphylococcus and Enterococcus cecorum, have been demonstrated to be pathogenic. You can isolate them from the bone lesions, you can grow them up in culture, and then my colleagues and people elsewhere in the United States have re-administered them to birds and shown that they do cause a reinfection. These are just some examples of the stages of what we're seeing, going from a normal, healthy proximal femoral head where we like to see this white cap of articular cartilage. This is the top of the growth plate. There's already an infection going on in this case. Then you can see damage to the bone and hollowing out of the bone with an abscess. Here we see a normal proximal tibial head. We see bacterial colonization. There's a colony there. And then we see gross infection here and clearly gross infection here. So these are examples of the bacterial infections that we're seeing. In order to study this, we've developed some mechanical models We don't want to inject pathogens into the birds or expose the birds to pathogens. We believe they're already present in the bird's intestinal tract, waiting for a wound site in the growth plate to be developed. So instead, we develop models such as these, in which the birds have to go up and over this little triangle that's made out of wire with an aluminum frame. And in doing so, that causes some footing instability. This footing instability basically creates osteochondrotic microfractures and triggers the onset of lameness. We actually call these devices speed bumps, familiar to most Americans as a way to slow down vehicles in the road. And I guess in South America they're called sleeping policemen or lazy policemen. But anyway, you get the idea. This is causing excessive stress and twisting and torque on the susceptible bones. So now to your question, can this be treated with antibiotics? And the answer is yes, and this has been the method of choice up until recently. This is an example using our model where the birds were basically exposed to these mechanical footing instability models. And the group that's showing up as blue circles here, going up to 40% cumulative lameness by almost 9 weeks of age, this group received only tap water to drink. The other group, shown in the red circles, began to exhibit lameness here, and on this day, they were administered enrofloxacin, which is one of the antibiotics of choice for treating this kind of a problem. The enrofloxacin stayed in their drinking water for over 2 weeks, and you can see that it brought the lameness to a halt. There was no further increase. But then bacterial resistance began to develop. That is, the enrofloxacin killed the susceptible bacteria, and the resistant bacteria took over. So this is creating bacterial resistance, and you can see the increase in lameness going on here. And then we stopped enrofloxacin in a withdrawal phase, and you can see the difference here. So basically, what we achieved with enrofloxacin was a 36% reduction in lameness. Although this is a severe challenge, you would never see this magnitude in the field. But the antibiotic clearly was successful, at least for a period of about 2 weeks. Now, the trouble with antibiotics is this development of bacterial resistance. And this is what we're trying to avoid with the use of non-antibiotic strategies such as probiotics to prevent this problem.
Yes, clear. Thank you very much. Before we go to the next poll question, I have one reader's question to you,
Okay.
Professor. Do you have any data on the prevalence of BCO in Asia?
So, as you can see from this figure, we typically don't see significant BCO even in our challenge model until about 30 days of age. That makes this an extremely important problem for people that are growing a large bird, say an 8-week, a 9-week, or a 10-week bird, where we're going from 3.8 to 5.2 kilograms in body weight. Nevertheless, in Asia, where we're growing maybe a 28 or a 30-day bird, we're going to have issues with BCO, maybe in a lower incidence of 1% or so, but also in product quality issues, where that white cap of articular cartilage separates from the femur head, and consumers reject that product. So we have subclinical issues, we have product quality issues, and we have a little bit of BCO mortality, usually starting about day 21, but certainly noticeable by day 28, when many birds in Asia are being processed.
Okay, let's see the next poll question in how our viewers see BCO lameness. The question is BCO lameness a common problem in your poultry operation? I will launch the poll for you, and you can vote by pressing A, B, or C. Yes, no, or not sure if there's a problem in your farm or operation. Some interesting results already coming in. It's almost a tie. Okay. I will show you. I will show the results. 38% of our audience say BCO lameness is a current problem in their operation. Is that something, Professor, you see in the field as well, these numbers?
So I've been speaking all over the world on this problem, and it's been remarkable. At the beginning of a presentation, people might believe or might claim that they're not seeing this problem. But in fact, almost every country I've been in Asia, Australia, Europe, North America, South America, and Central America, the pathologists clearly recognize that this is an issue. For example, one country I visited, there are multiple individuals claiming this is not a problem, and then it turns out that 40% of the mortality in broilers in that country actually are being culled because they're lame due to BCO. Mm-hmm. So my opinion is that wherever broilers are being grown, this is going to be a problem, and an important problem.
So everybody will experience it somewhere, sometime.
If you're growing modern broilers, you're going to see this.
Mm-hmm, mm-hmm. And what alternatives to antibiotics can be used to prevent and treat BCO lameness?
Okay, so this slide basically shows what we know as our pathogenesis, and where can we interdict this progression of events. What we've done is target gut health, because we believe that pathogenic species of Staphylococcus and Enterococcus and perhaps E. coli are harbored in the gut, maybe in small colonies, maybe in large colonies, we don't know, but they're gradually leaking through the intestinal epithelium. And it's those pathogens that make their way through the circulation to trigger these infections. If we can prevent or reduce pathogen leakage here, then we ought to be able to reduce the rate of bone infection. So our target has been on the improving intestinal health and improving the barrier function of the intestinal tract. So this is what the barrier looks like. We think much of this bacterial leakage is occurring across the small intestine. This is basically the upper end of the small intestine, and what we've done is evert the mucosa so that you can see the microvilli, or the villi. And here you can see the villi in a larger magnification, and now you can see the villi fully magnified, and you can see the vascular supply to the villi. And you can also see that these villi have microvilli. And look how delicate this tissue is. Normally this would be protected by an attached or adherent layer of mucus, but nevertheless, this is where the nutrients are going to be absorbed, and this is where the bacteria are going to be trying to leak in. So this is the key spot in the intestinal tract. This diagram shows the primary routes of microvilli or bacterial translocation. Basically, we have the enterocytes here. We have a mucus cell secreting the protective layer of mucus, the inner layer, which is adherent and relatively thick, and the outer layer, which serves as a lubricant to move the digesta along. And we hope to keep most of the pathogens in this outer layer or in the digesta and keep them moving along the intestinal tract. Some bacteria, such as Salmonella, make their way to the microvilli, bind to the microvilli, insinuate themselves into the cell, they can replicate in the cell, and then they exit at the basal membrane, are engulfed by macrophage and carried to the spleen and to the immune system to be neutralized or detected. The major route of pathogen entry seems to be between these tight junctions. Tight junctions seal the enterocytes to one another, but tight junctions can be penetrated by pathogens, and they too can be engulfed or escape and then circulate in the bloodstream. So this has been the focus of where we think we can have transcellular through the cell or paracellular between the cell bacterial leakage into the bloodstream. Now normally this seems to be a relatively slow process, but in response to stress and the production by the bird's adrenals of the stress hormone corticosterone, we get an inhibition of the immune system, and corticosterone can actually cause these tight junctions to open up. When that happens, bacterial pathogens can flood into the body, overwhelm the immune system, and cause polymicrobial bacteremia or sepsis. And clearly, these are going to make their way to the bones and cause an infection. So, how can we address these possibilities with probiotics? In this example, we're showing the likely roles of probiotics. In mammals, we know probiotics stimulate the enterocytes to produce defensins, which are natural antimicrobial proteins. We know the probiotics in birds can stimulate the immune system to respond or be sensitive to pathogenic bacteria. Probiotics can competitively exclude pathogens from binding to these microvilli and prevent, for example, salmonella from translocating through the cell. Probiotics can directly crosstalk through the microvilli to the enterocytes, cause the tight junctions to close up tightly, cause the nucleus to produce more messenger RNA for tight junction protein synthesis, and they also can stimulate mucus production to create this protective barrier that helps prevent bacterial leakage. So this is a biological rationale for using probiotics to prevent BCO in broiler chickens. We're trying to prevent or reduce both routes of pathogen entry through the epithelial layer of the intestine. This is an example of a doctoral thesis by Raj Murchison published in British Poultry Science showing these effects, or the key effects of probiotics. Basically, broiler chicks were raised for 3 weeks and exposed to reused litter. So this represents a natural challenge by exposing the birds to bacteria that are already present in the litter. And the birds were grown on a control feed or control feed containing probiotics. And then at 3 weeks of age, segments of the intestine were harvested and evaluated for permeability and mucus production. So in the bottom left-hand corner of this figure, we see the response of the transepithelial electrical resistance analysis. This basically says that if the tight junctions are closed tightly, then current will have a difficult time flowing through the epithelium, and so we'll have a high transepithelial resistance. If the tight junctions are open, we have a low resistance and current will flow easily between the tight junctions. And so we see a significant increase in tight junction integrity with a high transepithelial resistance for the probiotic strain. Another way of looking at this is to look at permeability to a large macromolecule. In this case, that macromolecule is bacterial lipopolysaccharide. It's part of the cell wall of gram-negative bacteria. And if the membrane has a low permeability to LPS, that means the tight junctions are closed. And we see a low permeability here compared with more open tight junctions with a higher permeability, statistically significant. And finally, the probiotics did stimulate mucus production by the enterocytes compared with the control group. So this is just physiological evidence that probiotics can do what we're saying they can do. Now we come to an actual series of experiments which were fairly remarkable because it's unusual to see this extent of success. This graph shows the results of 5 experiments, 1, 2, 3, 4, 5, conducted over a period of 2 years using 4 broiler lines, C, B, B, etc. So basically we have this first broiler line is very susceptible to BCO, and this last broiler line is genetically resistant to BCO. In all 5 of these experiments, the lowest incidence of BCO occurred when broilers were fed a control feed and grown on litter. So there's no or reduced mechanical challenge, and you can see in every experiment that that treatment is the lowest. Also in every experiment, the treatment with the highest incidence of BCO is when their legs were challenged mechanically with the wire barrier models, And we can see that the red bars represent the highest incidence of BCO in all 5 experiments. When they were in a similar mechanical challenge, but probiotics, Poultry Star was included in the feed, we got at least a 50% reduction in lameness, and sometimes it brought it down to levels that were not significantly different from the control level. Now remember that this is a severe challenge. Remember that enrofloxacin only reduced the incidence by 36%. So this is evidence of remarkable efficacy for this particular product. And to have this happen 5 times in a row over the course of 2 years with 4 different broiler lines is also remarkable. Now we have evidence that we can't show you from integrators in the United States and elsewhere that this probiotic is similarly effective in reducing the incidence of BCO lameness under normal commercial conditions of broiler grow-out. So one of the things that we have to warn people about is that not all probiotics are the same. This shows 2 experiments conducted under very similar conditions, but with a different probiotic. Here we have the level of BCO lameness with the control feed, and here we have it with the probiotic in the feed. In both of these cases, there was negligible response to the probiotic, and this shows both experiments combined. So this probiotic may work for some issues in gut health, but it certainly didn't protect against bacterial translocation that's responsible for BCO lameness. I should back up and say one more thing. In all of these experiments, we start the probiotic treatment on day 1, and we keep it in for the duration of the experiment. So that's been our challenge model in both of these experiments.
Well, thank you, Robert, for making this complex issue more clear to me and also for the viewers. I have one question from our online audience, and that is, what is the incubation time of the BCO infection?
So this is interesting. In some cases, we know the chicks are coming into the broiler house on day 1, and they're already contaminated with pathogens. We know this because if you harvest the spleen and culture the spleen, you can see Enterococcus in the spleen, you can see Staphylococcus in the spleen. Under those conditions, we typically still will not see BCO until 21 to 28 days of age, when they get enough weight on them to make some microfractures in the bones. My colleague at the University of Arkansas, Dr. Adnan El-Rubiai, has cultured some of these pathogens and administered them in the drinking water at 7 days of age. In those experiments, even after a pathogen was consumed by the bird for 24 hours, you see no response by the birds. The birds look clinically healthy. They grow perfectly well. For 4 or 5 weeks, you see nothing going wrong. And then suddenly you see BCO lameness. And it's much higher with a higher dose of the pathogen in the drinking water than it is with just drinking water alone. So we can say that the incubation time could well be short, but also there has to be some sort of body weight on the bird to initiate the microfracturing of the bone before we see this take off as BCO lameness.
So stress is more or less a trigger.
Right, exactly.
Weight stress. All right, thank you very much. That brings us to our next poll question, because we want to know what our viewers think and experience. The poll question is, have you used or recommended application of a probiotic to prevent BCO lameness in your operation? I will open the poll for you. You can click on the answer of your choice simply by pressing A for yes and B for no. I'll wait a couple of seconds for you to make your decision. It's looking good. Please enter your votes. And then I will stop and display the results to you. I see that 37% of you have used a probiotic to prevent BCO lameness. That's a decent score, but you are not all convinced yet. Luis, Poultry Star was developed specifically to address BCO lameness, or are there also other reasons to apply the product?
Well, I have different answers for this question, but yes, partially we kept in mind the barrier function, right? So we developed this product to reduce the failure, to reduce the barrier function failure. Okay, so in this regard, this brings me to, if I may, have this. And Professor Wideman, thank you for your presentation. It was highly knowledgeable. And here I have some of the key reasons to apply Poultry Star. I will show you something else later about the proven results we have, and these are peer-reviewed papers. But first, creation of eubiosis resulting in dry litter and improved production environment. Now, we have to remember in the composition, and this is something that I said before, we have different strains, right? So we try to cover throughout the whole digestive tract. And we have some strains from the cecum. These strains that were isolated from the cecum are also secreting short-chain fatty acids besides lactic acid. We have acids such as the butyric production in the right place. So this one will signal for the knockoff the agent that allows Salmonella to come to invade the small intestine. So because of this, we have suppression of the growth of commensal bacteria that is opportunistic, and then if it overgrows, it can become at some point something triggers it, as Professor explained, and it can become pathogenic. Then we consistently improve animal performance. This is not our main target, but by having the main drivers of gut health, and by having good gut integrity, we can improve performance, and we can have more uniform bird flocks. We can have a better animal response to vaccinations, and I will show you later the papers. We can reduce animal losses during the production cycles because we will have less mortality and no risk for animal or human safety, as I mentioned before. So what you can see there is a healthy gut. Well, we come from a disrupted microvilli or villi to a healthy villi by the use of the right probiotics. And here I was telling you about the proven results. We have results, or we have papers that have been published, and these are available online. Of course, they don't mention Poultry Star, but it says multi-species probiotic or multi-species symbiotic. We have against Campylobacter, Salmonella, and Clostridia. As Professor said, we have to mention that this is a preventive tool, meaning that you have to use it from day one, from the beginning, because it's difficult to control an onset situation when it's already chronic with a preventive tool, right? Sometimes this can work both ways. We have against Salmonella and Clostridia. We have also proven benefits for what we are known for, which is immunomodulation and microbiota. Here is important to mention that we have Poultry Star increased productivity index in broiler comparable to antibiotic growth promoter like flavomycin, and improve the IB, which is the infectious bronchitis vaccine, and the infectious bursa disease vaccination. So we improve the titers, and you can find all this documented online. And then also improve performance. You see there at the end it says that we were comparable, we improve performance if you compare our product with other growth promoters. And like I said, this is not our main target. Our main target is the immunomodulation, but because of this having a good gut integrity, then we can also improve performance. As a benefits, we will have the fast establishment of beneficial gut microflora, microbiota, meaning if you apply it from day one, reestablishment post-antibiotic applications. We can reduce the bacterial and enteric pathogens, gut integrity enhancement, and of course immunomodulation. So. that's what we had in mind when we created this product.
All right, well, thank you very much for explaining, Luis. Let's open our question and answer session, and please use the chat box to post your questions to our viewers. During the presentations, there were already a few coming in, so I will just have a quick look there. A question to Robert. How we differentiate BCO caused by bacterial translocation and spinal cord compression?
Aha. We include vertebral BCO along with femoral and tibial BCO. In other words, the evidence indicates that the vertebrae are being microfractured just like the growth plates in the leg bones. And so the pathogenesis seems very similar. In both cases, it may well be that birds come in as day one chicks already carrying pathogenic Enterococcus cecorum or Staphylococcus aureus, and it may take a while for this to develop into a lameness lesion, but basically the pathogenesis seems quite similar. The paralysis occurs when the lesion in the vertebral body gets so severe that the bone basically disintegrates. At that point, the body of the vertebrae collapses on the spinal cord and paralyzes the hind legs. That's a secondary response, though, to the initial bacterial infection.
To Luis, is your probiotic, or as you say, symbiotic, compatible with vaccines? I heard you mention the vaccines.
Well, yes, we have several papers. I was replying to one question today regarding these, where we have seen that we improved the response of the titers of the vaccines. So with the use of probiotics, and not only for the bronchitis or Newcastle disease, but also for Salmonella and for other type of vaccines. Now we just have to be careful on how do we apply it. So in this regard, if somebody wants to use Poultry Star with a vaccine, I will suggest you to contact us, and then we will give you the best recommendation. Yes.
And also about the product, another question from our audience. Do you have data on Poultry Star in pullets?
Yes, we have. We have information, and I'll be glad to share it through emails if they have a more direct question. But yes, we have. We can use Poultry Star. We have seen that we can use Poultry Star up to before laying, and then you can see during the laying period a more stable production of eggs, and also sometimes it also helps in the formation of the integrity of the eggshell. Yeah. Yes.
I see a lot of questions rolling in. We'll make a selection. I have one for Professor Wideman. What could be the specific or possible reason that some broiler farms seem to be more susceptible for BCO, especially through Enterococcus cecorum, compared to other farms? This is an interesting question, and if somebody knows an answer that they can prove, I'd be very interested in hearing it. Not to be facetious, but in every country that I've visited, the term repeater farm is very familiar. And that means that regardless of the hatchery, regardless of the feed source, these farms tend to be prone to outbreaks of BCO. In multiple farm situations, the problem tends to be that, or multiple house on the same farm situation, the problem seems to show up in one house for one flock cycle and another house in another flock cycle. So why is this a repeater farm if it's not showing up in the same barn flock cycle after flock cycle, but it's showing up in some of the barns on that farm? We think that there are levels of stress. There may be levels of biofilming of pathogens in the drinking water. And frankly, we don't have all the answers. One interesting observation is that when a barn breaks with BCO, typically the earliest lame birds are found within one local area. And then gradually the lame birds are being picked up by the grower or the farmer in a broader and broader area. So this could indicate that in certain barns or where certain chicks are placed, we have a low-level vertical transmission or a low-level environmental contamination that then starts the process, and as the birds shed the pathogens, it spreads horizontally. But that would also be addressed by keeping the probiotics in the feed because you can reduce that horizontal spread. I would say over half of the poultry farms I've visited, if you ask the person, where did you first start picking up lame chickens, they can point to a very specific spot in an affected barn. Okay.
And how significant is the role, it's another question, of E. coli in causing BCO? And are there any experiences from experimental studies, is the question.
Yeah. So our team from the microbiology group at the University of Arkansas in the biology department and others have also isolated E. coli from these lesions. And many of these E. coli have been typed. The issue is, I don't know of any experimental evidence where those isolates of E. coli have been reintroduced into naive birds and have been demonstrated to trigger similar lesions. That kind of study has been done with Staphylococcus ignitis, for example, at the University of Arkansas, and Enterococcus cecorum at North Carolina State University Veterinary School. You can isolate those pathogens, put them back into birds, and reintroduce these lesions. But that hasn't been demonstrated for E. coli. So it may be that the E. coli are just there and they're being isolated, or it may be that they have pathogenic potency, but we don't know the answer to that. It has not been demonstrated experimentally, to the best of my knowledge.
Clear, so further research needed there.
Further research is needed.
Can we use probiotics when the birds are already infected with BCO?
So there's two levels of thinking about this. One level is we did an experiment where we waited with our mechanical challenge until day 28 when the birds first started exhibiting lameness. At this stage, they've already been stressed, their legs have been stressed, and they've been stressed by the mechanical challenge. They've already leaked bacteria into the bloodstream, it's already started to infect the bones. And in that situation, where we came in and tried to treat the problem therapeutically by administering probiotics, as if you were treating it therapeutically with an antibiotic such as enrofloxacin, we did not have success. We basically say that if you're going to prevent bacterial translocation, you need to do it as early as possible. You need to start at day 1, and people are, BIOMIN and others, but BIOMIN in particular is developing a system for actually spraying droplets of probiotic onto the chicks at the hatchery to try to get it into the chicks as soon as possible to stop this horizontal spread. The other issue with an existing infection is sometimes these existing infections occur or are evident in newly hatched chicks. And in that case, if you have a low level of infection, vertical transmission or hatchery infection, you want to prevent those newly hatched chicks from spreading that. So even though they're infected and they may go ahead and get BCO lameness as the flock cycle progresses, if you can prevent them from shedding and transmitting that horizontally to their flock mates, that's a good use of probiotics. So you can't use it as a therapeutic treatment, but you can use it as a prophylactic treatment and to prevent horizontal transmission.
Right, clear. When it comes to administering the probiotic, can you tell something about that, Louise? And the question is, what about heat stability of the poultry star product?
That's a good question. It come up a lot of times. People is concerned about heat stability. Well, the reason is because lactobacillus are very susceptible to this, and when we compare the difference between bacillus-based product, they are spores, right? So they are very stable. However, we have developed our own technology where we microencapsulate all these bacteria, but individually, and then we mix them in the ratios that we in our own formulations, and then we can secure, or we can assure that we have good heat stability. up to 85% during 90 seconds, 85% degrees Celsius for 90 seconds, there's no problem. You will have 100% of recovery. However, if you go up to different, very harsh conditions of pelleting or preconditionings we have had cases of to 90, no, 80, it was 85, no, it was 90 degrees Celsius, and it was for 6 minutes, so it's a very harsh condition, and we still have a good recovery of 78% with the technology of microencapsulation. We also have the soluble version, but this one is more for the use in the water. So yes, we are stable, we have heat stability, and we have proven the data.
Yeah. Yep. So no issues there in the feed mill?
No.
Yes. To Professor Wideman, do you think all probiotics are as effective to reduce BCO lameness in poultry flocks?
Well, the answer to that is definitely not. And I guess I can't point to the exact reason for it, but Poultry Star, for example, is definitely effective. We've tested in our research model another probiotic which has some positive response or some efficacy, and then we've tested probiotics that have no positive response, no efficacy. So the nice thing is that when they show up in our research model to be efficacious, as Poultry Star has, then when you put them into a real-world broiler grow-out situation, they're also efficacious because we're exhibiting or exerting an extreme rigorous challenge. And so under less challenging conditions, they definitely shine. The other evidence is those that don't work out well for us, at least from one integrator, was also the probiotic that didn't help his situation at all. So we believe we can predict, although it's a 8-week challenge trial, which is very labor intensive and very expensive, at least in our model, that translates well. If it's efficacious in that trial, we're going to see a positive response in the field. If it doesn't work in that trial, we probably won't see much, if anything, going on. So they do differ. They're not all the same. My opinion is that it depends upon how well they interact in the small intestine, particularly the lower two segments of the small intestine.
I've selected one more question from our viewers. Is probiotic with Bacillus strain, or can it be used to reduce BCO prevalence?
Luis, I'm going to defer to you on that one.
Yes, well, we don't directly test a competitor's product. However, through data that is available online as well, we have seen that sometimes these bacillus are not as effective as our strains, and the reason is because what I was telling you before. If we put the categories, we are immunomodulators, and then we tighten up, as Professor Wideman, we have to tighten up these tight junctions, okay? So in bacillus products, they are good for growth performance. They are good probiotics, I won't deny that. That's their main target, growth performance, because they can secrete enzymes that can help into the breakdown of nutrients, and then there's a better absorption of the nutrients, right? However, and they have a secondary effect. They will have the promotion of lactobacillus growth, because they saturate the system with bacillus products or bacillus strains, and then these lactobacillus can grow. So indirectly, they will have the effect as well. But if you think about it, why don't you just invest a little bit more, and then you can provide the direct drivers of gut health, and then you will have the first effect. You will prime the immune system, you will have good gut integrity, and after that, you will have good performance. So I think that's what we can answer in this regard, if Professor Wideman agrees.
I agree.
Well, it's nice to have everybody agree on this on this table. Well, that brings me to the end of the questions which are posed online. Well, thank you very much, Louise, and also Robert, for your presentations and your patience in answering all my questions and of course the viewers' questions. That brings us already to the end of this webinar on antibiotic-free solutions to control BCO lameness. Thank you for joining us. One last remark when I can get to the next slide. If you want to learn more about this subject or would be contacted by a BIOMIN representative, please press the button which is on your screen now. I also kindly request you to give us feedback on this webinar and its contents by completing the questionnaire which will be presented to you after our live broadcast. Thank you again. Thank you guys.
Thank you.
See you next time.
Thank
Thanks.
you.